Welcome to G Fun Facts Online. Today, we are discarding the old map of environmental health and drawing a new, far more unsettling one. In the grand theater of human health, the lungs have long held the spotlight as the primary victims of air pollution. For decades, the narrative was clear. We believed that smog chokes the breath, soot blackens the alveoli, and the cardiovascular system labors under the strain of oxygen deprivation. Yet, a silent and far more insidious invasion has been taking place, one that bypasses the chest entirely to strike at the very seat of human consciousness. We are witnessing a paradigm shift in environmental medicine. The frontier of toxicology has moved upstream, from the respiratory system to the central nervous system. This is the era of neurotoxicology, a discipline revealing a terrifying truth, that the air we breathe is reshaping the architecture of our minds, dismantling our memories, and accelerating the decay of our neural networks. The link between particulate matter and neurodegeneration is no longer a hypothesis. It is a biological reality documented in the autopsied brains of urban dwellers, in the inflamed hippocampi of laboratory models, and in the epidemiological data of millions. From the traffic-choked arteries of megacities to the smoke-shrouded valleys of wildfire country, we are inhaling a cocktail of neurotoxins that threatens to spark a global pandemic of neurological decline. To understand the magnitude of this threat, we must first appreciate the scale of the enemy. Particulate matter is not a monolith. It is a complex, size-dependent hierarchy of destruction. While PM10 or coarse particles might irritate the eyes and throat, the true assassins are the fine particles known as PM2.5 and the ultrafine particles known as UFPs or PM0.1. PM2.5, with a diameter less than 2.5 micrometers, is small enough to penetrate deep into the lung tissue and enter the bloodstream. But PM0.1, nano-sized particles often smaller than a virus, possess a terrifying mobility. They do not merely enter the body, they permeate it. Current research identifies three primary highways utilized by these particles to breach the brain's defenses. The first highway is the olfactory bypass, or the direct route. You see, the nose is more than a filter, it is a direct conduit to the brain. The olfactory bulb, located just above the nasal cavity, is the only part of the central nervous system directly exposed to the external environment. When we inhale heavily polluted air, nano-sized particles laden with heavy metals like magnetite and combustion byproducts deposit on the olfactory epithelium. Unlike other nerves, olfactory neurons lack a protective myelin sheath at their terminals. These particles are swallowed by the neurons and transported via retrograde axonal transport directly into the olfactory bulb. From there, they have an unimpeded path to the entorhinal cortex and the hippocampus, which are the exact regions where Alzheimer's pathology begins. This nose-to-brain highway allows toxins to bypass the body's primary filtration systems entirely. The second pathway is the Trojan horse, or the systemic route. When PM2.5 crosses the alveolar capillary barrier in the lungs, it enters the bloodstream. Under normal circumstances, the brain is guarded by the blood-brain barrier, a fortress of tightly knit endothelial cells that strictly regulates what enters the central nervous system. However, systemic inflammation caused by lung irritation releases a flood of cytokines, specifically IL-6 and TNF-alpha, into the blood. This chemical assault weakens the tight junctions of the blood-brain barrier, making it leaky. Once the gate is compromised, particles circulating in the blood, or immune cells that have engulfed these particles, slip through the defenses and enter the brain parenchyma. The third pathway is perhaps the most surprising. It is the gut-brain axis. Emerging research suggests that inhaled particles are often cleared from the lungs via mucociliary escalators, swallowed, and end up in the gut. There, they alter the gut microbiome, causing dysbiosis and intestinal permeability, also known as leaky gut. This local inflammation can travel up the vagus nerve, a primary information superhighway connecting the gut and the brain, triggering a neuroinflammatory response deep in the brainstem. Once these particles embed themselves in neural tissue, they do not simply sit inert. They are chemically active landmines. The composition of particulate matter varies. Diesel exhaust particles are rich in polycyclic aromatic hydrocarbons, while industrial dust may contain neurotoxic metals like lead, manganese, and iron. The damage occurs through a mechanism known as the toxicological triad. Phase 1 is oxidative stress. Many particulates, particularly those containing transition metals like iron and copper, participate in Fenton reactions, generating free radicals. These reactive oxygen species attack the lipid-rich membranes of neurons. The brain, which consumes 20% of the body's oxygen, but possesses relatively low antioxidant defenses, is uniquely vulnerable. This oxidative stress damages mitochondria, the energy powerhouses of the cell, leading to energy failure and neuronal death. Phase 2 is microglial activation, or neuroinflammation. Microglia are the brain's immune sentinels. In a healthy brain, they are housekeepers. pruning synapses and clearing debris. However, upon detecting foreign particulate matter, they shift into an aggressive state known as M1. They begin to release a storm of pro-inflammatory cytokines and neurotoxins intended to destroy the invader. But because the particles are inorganic and cannot be degraded, the microglia remain perpetually activated. This chronic neuroinflammation becomes a scorched earth policy, killing healthy neurons alongside the invaders. This state of chronic glial activation is a hallmark of every major neurodegenerative disease. Phase 3 is protein aggregation. Perhaps the most chilling link is the interaction between air pollution and the specific proteins associated with dementia. Studies have shown that oxidative stress and inflammation impair the brain's glymphatic system, which is the waste clearance system active during sleep. Consequently, toxic proteins like beta-amyloid and tau, which are associated with Alzheimer's, and alpha-synuclein associated with Parkinson's, fail to clear. Furthermore, certain particles can act as seeds or nucleation sites, physically accelerating the clumping of these misfolded proteins. We are essentially inhaling the catalysts for plaque formation. It is also important to note that the source matters. Not all smoke is created equal. While the mechanisms of damage are shared, the chemical payload differs by source. Historically, diesel exhaust particles have been the most studied neurotoxins. Rich in carbon and coated with organic chemicals, they are uniquely effective at disrupting the blood-brain barrier. Living near major roadways has been consistently associated with lower cognitive scores and higher dementia rates. The magnetite theory is particularly disturbing here. Researchers have found millions of magnetic nanoparticles in the brains of urban residents, particles that match the high-temperature combustion profiles of vehicle engines, distinct from naturally occurring iron in the body. However, in recent years, a new and more potent threat has emerged: wildfire smoke. As climate change turns forests into tinderboxes, the smoke generated is not just burning wood, it is burning structures, plastics, insulation, and chemicals. Recent comparative toxicology studies suggest that wildfire smoke may be up to 10 times more neurotoxic than traffic pollution. The particulate matter from wildfires is often more oxidative and inflammatory, likely due to the complex mixture of burning biomass and anthropogenic materials. A 2024 study analyzing millions of health records linked wildfire PM 2.5 exposure to a significantly higher risk of dementia diagnosis compared to equivalent levels of non-wildfire PM. Furthermore, the newest frontier in neurotoxicology is the study of nanoplastics. We have long known these particles permeate our oceans and food supply, but we now know they permeate our minds. Research from 2024 and 2025 utilizing advanced spectroscopy has detected skyrocketing levels of plastics in human brain tissue samples, up to 50% more than samples from just a decade ago. These lipid-soluble plastics can hijack fatty transport mechanisms to cross the blood-brain barrier with ease. Once inside, they may physically disrupt cellular machinery or leach endocrine-disrupting chemicals directly into the neural environment. The clinical outcome of this toxic accumulation is a broad spectrum of neurodegeneration. The link between PM 2.5 and Alzheimer's disease is the most robust. Pollution exposure is associated with increased amyloid plaque burden, even in young adults and children in highly polluted cities like Mexico City. The risk is particularly pronounced in individuals carrying the APOE4 gene, suggesting that air pollution acts as a second hit that tips genetically vulnerable brains into active disease. Regarding Parkinson's disease, it is often described as an environmental disease with a genetic component. The loss of dopaminergic neurons in the substantia nigra is highly sensitive to oxidative stress. Heavy metals found in industrial pollution specifically target the basal ganglia. Epidemiological hotspots for Parkinson's often align perfectly with maps of industrial particulate emission, particularly in the Mississippi-Ohio River Valley and the Rust Belt. And the damage is not limited to the elderly. In children whose blood-brain barriers are still developing, exposure to high levels of traffic pollution is linked to reduced white matter volume, lower IQ, and higher rates of ADHD and autism spectrum disorder. We are potentially compromising the cognitive capital of entire generations before they even reach adulthood. The picture painted by neurotoxicology is grim, but it is not hopeless. Understanding the mechanism of injury provides us with a roadmap for defense. If the pathway is inhalation, leading to inflammation, leading to neurodegeneration, then we can intervene at each step. The first line of defense is filtration and avoidance. High-efficiency particulate air, or HEPA filters, are not just luxury items. They are essential medical devices for brain health. Studies show that using HEPA filtration in homes can lower intracellular inflammatory markers within weeks. On high pollution days, specifically during wildfire events, N95 respirators are the only masks capable of blocking the ultrafine particles that threaten the brain. Beyond filtration, we can fortify the brain's internal defenses through dietary interventions. Research indicates that high systemic levels of omega-3 fatty acids, specifically DHA and EPA found in fish oil, can attenuate the neurotoxic effects of PM2.5. They act by resolving inflammation and maintaining the structural integrity of the blood-brain barrier. Another powerful tool is sulforaphane. Found in broccoli sprouts, this compound is a potent activator of the NRF2 pathway, which is the body's master switch for antioxidant production. Unlike standard antioxidants that neutralize one free radical at a time, sulforaphane triggers the expression of enzymes that can neutralize millions of free radicals and aid in the excretion of airborne toxins like benzene. Additionally, supplementation with B vitamins, specifically B6, B12, and folic acid, has been shown in some trials to mitigate the epigenetic damage caused by air pollution, essentially preventing the switching off of genes that protect our immune system. Ultimately, personal protection is a stopgap. The solution lies in policy. We must reframe air quality regulations not just as respiratory protection, but as dementia prevention. Costs associated with Alzheimer's and Parkinson's care are astronomical, meaning that reducing PM2.5 levels is likely one of the most cost-effective public health interventions available to reduce the burden of these diseases. We are standing at a precipice. The industrialization of our atmosphere has collided with the biology of our brains. The particles we release from our tailpipes, smokestacks, and burning forests are not disappearing into the ether. They are accumulating in the synapses of our collective consciousness. Neurotoxicology has illuminated the invisible link between the smog outside and the fog inside. It challenges us to look at the rising tide of neurodegenerative disease, not merely as an inevitability of aging, but as a consequence of our environment. The air is toxic, but the knowledge is curative. By acknowledging the link between particulates and neurodegeneration, we arm ourselves with the motivation to clear the air, not just for the sake of our lungs, but for the salvation of our minds. Thanks for listening to G Fun Facts Online.